Bodies of living organisms contain large number of different molecules which constitute their structure. They are also part of various physiological processes taking place in primary structural materials of organisms are proteins and cellulose. By means of the unique process of photosynthesis, plants produce carbohydrates. Plants utilize the minerals absorbed by their roots to produce proteins. Lipids are the main ingredient of vegetable oils and milk fats. Nucleic acids constitute the genetic material of organisms. What are carbohydrates? Carbohydrates are the polyhydroxyaldehyde, or polyhydroxyketone, or the compound on hydrolysis produce. Polyhydroxyaldehyde or polyhydroxyketone. Some carbohydrates like glucose, fructose are sweet in taste and are called sugars. The most commonly used sugar is sucrose, which is obtained from sugarcane or sugar beet. The sugar present in milk is called lactose.
With the classification of carbohydrates based on the products of hydrolysis, they are classified into two different categories. The first one is simple and the next one is complex carbohydrates.
Monosaccharides do not hydrolyze further into smaller units of polyhydroxyaldehydes or ketones. Here the example of monosaccharides is glucose. Here glucose do not undergoes hydrolysis to give any other polyhydroxyaldehyde or ketone. So glucose is example of monosaccharides, it is a simple sugar. Another example is fructose, ribose. Complex is further divided into oligosaccharides and polysaccharides.
Now, what do we mean by oligosaccharides and polysaccharides? Let's see. Oligosaccharides means oligo is equal to few, saccharide is equal to sugar molecules or monomers. And few means how much? The few means 2 to 10 monomers.
Oligosaccharides on hydrolysis yield 2 to 10 units of monosaccharides.
Oligosaccharides further are classified as disaccharides, which are compounds which on hydrolysis gives 2 sugar monomers. Example for this is sucrose, which is made from glucose and fructose. The next example is maltose, made from glucose plus glucose. The next is lactose, made from glucose and galactose. Moving further, oligosaccharides are divided further into trisaccharide as well, which are nothing but compounds which on hydrolysis gives 3 sugar monomers. And these compounds have a general formula as C18H32O16. Example for this is raffinose, which comprises of glucose, fructose and galactose. Similarly, there is one more division called as tetrasaccharides. What are tetrasaccharides? As the name suggests, these are compounds which on hydrolysis gives 4 monomers or 4 sugar units. And these have a general formula as C24H42O21. Example for this is statiose, which comprises of glucose, fructose, and 2 molecules of galactose. Poly means many, and saccharide means again sugar or sugar molecules. carbohydrates, which on hydrolysis will yield a large number of monosaccharide units. And the example for this are nothing but starch, cellulose, glycogen, etc. In lab method, for the preparation of glucose, what do we require? We require a molecule of sucrose, and if it is sucrose, the method has to be a lab method. Let's start. Sucrose, when boiled with dilute acid, gives glucose and fructose. Let's see the reaction. We have a molecule of sucrose plus water. In presence of an dilute acid like HCl or H2SO4 and heating, what do we get? We get two different products. We get first that is glucose and the next that is fructose.
Separation of glucose and fructose from their mixture. Mixture of glucose and fructose is taken in 90% ethanol alcohol and heated below 323 K. Glucose is separated from fructose during cooling. Glucose being almost insoluble in alcohol crystallizes out first. The solution is filtered to obtain crystals of glucose.
Let us see the basics part of glucose. How does a glucose structure look like? So, open chain compound of glucose. So glucose has how many carbon atom? Glucose has 6 carbon atom, and it is all are placed in a straight chain. So here there is a glucose structure. You can see here there is 6 carbon atoms, and there is here you can see here I have written a OH should be on right hand side, two right hand side, on left side and right side. What is the meaning of this? This is the OH, this one is OH. This OH is this one. Now bring then attach OH on the right hand side. So I am attaching OH on the right hand side. Then again you attach right hand side, you attach OH. Okay. Now you attach OH on the left hand side. So here we will attach OH on this side. Now we have to attach the OH on the right hand side. So here we will attach. And the rest remaining you have to attach hydrogen. So hydrogen, hydrogen, hydrogen and hydrogen. So this is the structure, this is the open structure of a glucose. So this is the actual glucose structure looks in open chain compound. So what is the explanation? Glucose has 6 carbon atoms. So you can see 1, 2, 3, 4, 5, 6. 6 carbon atom in glucose, and it is all the carbon atom are in the straight chain. Okay, next point, glucose has aldohexo structure. So here you can see this CHO. This CHO is a aldehydic group. You know that CHO is a functional group of aldehyde. So glucose has one aldehydic group. And how many carbon? See, here it is written aldohexo. Aldo means aldehyde. What is the meaning of hexo? Hexo means 6 carbon atom. Here you can see 1, 2, 3, 4, 5 and 6. So there are 6 carbon atom. Therefore it is named as hexo. So totally what we have named, since there is a aldehyde group, it is known as aldo, and there is 6 carbon, it is known as hexo. So glucose is a aldohexo structure. Next, glucose molecule contain one aldehydic group. So you can see in whole structure there is only one aldehydic group, and it is known as formyl groups. CHO is known as formyl group. And the remaining 5 carbon atom carry one hydroxy group. Except this, remaining all the carbon atom has one OH group. See, second, third is OH, fourth is OH, fifth is OH, sixth is OH. So all have except CHO, all carbon atom has OH group. So there are 5 hydroxy group.
Reaction to prove straight chain structure of glucose. Here no of carbon atom present in glucose is 6. And it is straight chain.
Reduction of glucose. Nothing but glucose on treatment with HI. So here I have a molecule of glucose which is treated with HI for 2 to 3 hours. And in presence of heat, What exactly I obtain is an alkane, that is N-hexane, which proves that glucose consists of 6 carbon atoms and they are arranged in a straight chain. Next reaction. The second reaction was reaction of glucose with hydroxylamine and hydrogen cyanide. Let's see for this. We have a molecule again of glucose. This molecule is treated with NH2OH, that is hydroxylamine. What would happen is H2 and O combines to give us water. So on the arrow we would write minus H2O. Let's see next. Now the groups which are remaining combine together to give us the corresponding oxime, and therefore this molecule is now called as glucoxime.
Glucose when treated with hydrogen cyanide. There is a breaking bond between the C and O. And there is a single bond between CH and O. So now, CN will go to carbon atom and thus there is attachment of CN group and hydrogen will attach to oxygen atom. And this result in formation of OH. Thus result structure formed is glucose cyanohydride.
Reaction which we studied was glucose on treatment with bromine water, a mild oxidizing agent. Remember students, let's see the reaction for this. We have glucose treated in presence of bromine water. What we get? Now this group, that is an aldehyde group, gets converted into C double bond O OH, and therefore this compound is now known as gluconic acid, symbolizing that we have a aldehyde group.
Further, the fourth reaction, that was glucose plus nitric acid. Let's see what happens. We have a molecule of glucose. To this, we are adding dilute HNO3. Now, again, let's see. First of all, the aldehyde group, that is COOH, would get converted into C double bond OOH. Similarly, the primary alcohol would also get converted into C double bond OOH. So CH2OH gets converted into C double bond OOH, that is carboxylic acid group. And the name of this compound is
saccharic acid,
symbolizing that yes, you have a primary alcohol present into glucose.
Test to prove that glucose contains 5-hydroxy group when treated with acetic anhydride. So this is the glucose which is reacting with 5 molecule acetic acid anhydride. So this part is known as acetate group. So this acetate group I have removed, okay, and from the glucose below you can see this one, this part, CH2OH part. Here the hydrogen will be removed. So I am removing the hydrogen and it is attaching with the acetate group to form acetic acid. Formula is CH3 C double bond dash OH. Like that, second molecule, here hydrogen is removed. Now the hydrogen is removed from the here CH dash OH. One hydrogen is removed to form a second molecule of acetic acid. Like that, third molecule acetate ion, hydrogen is removed, acetic acid is formed. Another molecule of acetate ion, hydrogen is removed to form acetic acid, and a last molecule acetate ion and one hydrogen atom. So all the hydrogen atom is removed, 4 from this and 4 from here. Total 5 hydrogen is removed and it get attached with the 5 molecule of acetate ion to form a 5 molecule of acetic acid. 1, 2, 3, 4, 5. So there is a formation of 5 acetic acid. Okay, and the remaining part, this is CH3C double bond O, this is the remaining part, acetyl group. So this acetyl group, what I will do, just I will adjust it and make it like this, dash C double bond O dash CH3. And what will happen to this part, how many molecule of this part is there, CH3C double bond, there are 5 molecules remaining. So what will happen, this CH3C double bond O will going to attach out of this 5 CH3C double bond, the 4 will go here it will be attached with, and 1 molecule will attach here. Okay, so this is the part where H is already removed, where to give acetic acid. So this part is remaining here, that this part will going to attach in here, 4 molecule here and 1 molecule here. So this will go and attach below part. Okay, the 1 molecule is attached. I mean, remaining there is 4 CH3C double bond is remaining. That 4 CH3C double bond will go here in the middle part. So this part is gone here. Okay, so total 4 molecule is attached here and 1 molecule is attached here. Total 5 molecule is attached, and this type of structure is known as glucose pentacetate. So this glucose has how many OH group? 5 OH group is there. So how many acetate group is attached? 5. So what this indicate, that this 4 it indicates that because of the formation of pentacetate, so it indicates that the glucose has 5 OH group. More if suppose it was 6 OH, then it was 6 acetate group was attached here, but here there is 5 OH group. That indicates that that glucose has 5 OH group, 5 hydroxy group is attached. The formation of glucose pentacetate indicates that glucose has 5 OH group, that is hydroxy group.
Thank you for watching this video. If you like this lesson, please like, share, and subscribe to Aklesh Sir AV. Click on the next video to continue learning. See you in the next class.